Let's now dive into the mechanisms behind autoimmunity. And we have to start by going back to where we talked about tolerance, way back when we looked at how B cells and T cells develop and mature. We talked about a lot of different mechanisms that the bone marrow and the thymus both had in preventing self-recognition or preventing B cell receptors or T cell receptors that recognize self-antigen. We looked at negative selection in the bone marrow for B cells. We looked at negative selection in the thymus for T cells. We looked at Treg suppression out in the periphery for secondary tolerance. We talked about how there's different immune privileged areas in the body where there's a lot of decreased immunity and low inflammation, like in the brain, in the eye. And then we also talked about how if a T cell or B cell gets out into the periphery and does recognize self-antigen, they can be shut down or rendered anergic. So we had peripheral tolerance, we had central tolerance, we had immune privilege, all of these different things that help prevent an autoimmune disease from occurring. But we do see that sometimes, on rare occasion, that self-tolerance at some point along the way is lost, or a B cell or T cell escapes, likely a T cell, and inadvertently, there is a reaction or recognition of self-tissue. And so what happens then is we have an immune response against normal, healthy self-tissue. The immune response, like any good immune response, is going to try to clear all of the dangerous antigen. But in this case, it's not dangerous, it's actually healthy, but the immune system doesn't know that. And so it sees its target and it's going to try to clear its target until A, the antigen is completely wiped out from the body, or B, the patient dies because of the effect on the tissue. And so there's this chronic stage of inflammation that occurs throughout the individual's life who have this type of interaction going on. This is going to lead to decreased function of the tissue and an environment where more tolerance can potentially be lost because of a high inflammatory environment that's being produced. And so this can actually increase in breadth and strength over time. So although these regulatory mechanisms have been put into place, there can be temporary relief. Like maybe the Tregs can come in and say, okay, let's shut her down, but not everybody goes to sleep. And so autoimmune diseases, although can kind of go up and down, you can relapse, you can have, peaks, you can have, low times. Rarely ever are autoimmune diseases cured or fully resolved. They can be kept at bay. And these are chronic diseases that are going to kind of just be there for the entirety of that person's life. And then as inflammation occurs, then the body repairs and there's reconstruction, and then there's destruction and reconstruction, which can alter the tissue as well. But largely, these are lifelong chronic diseases that people have to manage because there really is not a cure. There was this one gene, though. Oops, I thought I was on pen here. AIR. Remember that protein or that gene that we talked about a long time? It was a transcription factor. Sorry, not a protein. Transcription factor way back when we looked at T cell development. And we talked about how thymic epithelial cells have the ability to turn on transcription of literally any gene of the body. So any gene can be transcribed, meaning that even tissue-specific proteins within the thymus tissue-specific out in the body can be produced by these thymic epithelial cells to train or to catch T cells that could potentially recognize that self-antigen. And we see that a lot of people that lack AIR transcription factor also have autoimmune issues. And so this is very compelling evidence for the connection between tolerance and autoimmunity. And we see that susceptibility to autoimmune diseases typically runs in families. It can be genetic, it doesn't have to be. We also see that it differs between populations and ethnic groups. And so genetic factors do potentially play a role in a lot of autoimmune susceptibilities. It seems like HLA is really important for autoimmune heredity. And so in almost every autoimmune disease, there seems to be some sort of HLA association. And autoimmune diseases tend to be more prevalent in people from European descent, white individuals, rather than from other groups of people. And so there is a haplotype, an HLA haplotype that I have there that is often associated with a higher frequency of autoimmune diseases. There's also going to be prevalence of autoimmune diseases across genders. So females tend to have significantly more autoimmune diseases than men do. And in general, we kind of can link it to sex hormones. Women tend to make a stronger adaptive immune response to regular infections than men do. And this could potentially be because of the estrogen, prolactin, and testosterone differences between males and females. But if you look at this table, there's only one autoimmune disease that we see more often in men than we do women. And all of these diseases we see more often in women than we do men. So there definitely is a propensity of women to have autoimmune diseases over men. If we go back to the idea with HLA being important in autoimmunity, we have to think about HLA and what they code for. We know we have class 1, we have class 2, and they code for our MHC molecules. And our MHC molecules are vitally important. They're on pretty much any nucleated cell of the body, HLA1 is, and HLA2 or MHC class 2 are going to be on those antigen-presenting cells. But because some antibodies or some autoimmune diseases are mediated by antibodies and some are cell mediated, we can kind of see that there's some sort of association between MHC class 1 versus MHC class 2 when we look at strength of autoimmunity. And we see that the number, the nature, and the strength of the disease really has everything to do with T cells. B cells, yes, have MHC class 2 on them, and yes, they're involved in T cell presentation, but T cells run the show from the beginning. And the fact that a single HLA allotype is associated with disease suggests that if a T cell is going to recognize that HLA, it's the T cell that at some point loses tolerance. And any T cell that is a clone of that initial self-recognizing T cell will also recognize self-antigen. And so loss of T cell tolerance really is the necessary first step in any type of autoimmune disease. Now, you may say that some autoimmune diseases are antibody driven, right? We looked at a couple of them in the previous lecture, but we know that B cells can't make antibody without the help of T cells. And any autoimmune disease that is driven by antibody is going to be driven by antibody that is isotype switch and has a high affinity towards antigen, meaning a T cell or a T follicular helper cell at some point had to interact with that B cell, meaning that T follicular helper cell has to recognize self-antigen, meaning that naive T cell, where it came from, somehow escaped tolerance or negative selection or some sort of T cell tolerance loss. So it all comes back to T cells losing their tolerance first. Mechanisms for eliminating autoreactive cells from the naive B cell population, they're there. We have negative selection for B cells, but it's far less efficient than what we see with T cell negative selection. B cells go through negative selection in the bone marrow, and the only proteins they'll be exposed to or antigens they'll be exposed to in the bone marrow are those that are present in the bone marrow. Whereas T cells, because they go through negative selection in the thymus, they have the additional set of tissue-specific proteins being presented to them via the AIR transcription factor. And so T cell negative selection is arguably much better and stronger than B cell regulation, tolerance, negative selection. So that makes sense that the body is going to put more effort into preventing T cells from escaping that could recognize self, because really, even if a B cell gets out of the bone marrow and recognizes self-antigen, it's not going to be able to make self or antibody against self until it has a T cell that also recognizes self. So just, make sure the T cell doesn't get out. So there are many more autoimmune diseases that are associated with MHC class 2 than there are MHC class 1. And that then tells us that CD4 positive T cells play a larger role. They're more likely to lose tolerance or escape tolerance than a CD8 positive T cell, which, thank goodness, because a CD8 positive T cell can recognize MHC class 1, which is on every nucleated cell of the body. But that also points to the reason that there's such a higher mountain that the CD8 positive T cell has to overcome, that naive CD8 positive T cell has to overcome in order to be activated. Oh my gosh, we just put a whole bunch of stuff together there, you guys, a whole bunch of stuff from previous chapters, bringing it forward. A CD8 positive T cell is going to recognize MHC class 1, but it has to overcome CD4 positive T cell help, usually, to get activated. And so we don't see as many CD8 positive T cell driven autoimmune diseases because there's a really, really high protection and a lot of effort put into maintaining CD8 positive T cell tolerance, which is good. We don't want to destroy all the tissue cells of the body. CD4 is only, or MHC class 2 is only located on a subset of cells. Okay, I hope that was okay. I hope I muddled my way through that. You can always ask me questions if you need. Several autoimmune diseases are caused by antibodies, and antibodies that are directed towards self, we call them autoantibodies. And those autoantibodies can bind to a cell, typically like a receptor or something, and either turn it on or turn it off, activate it or block it, right? And so we use the terms agonist and antagonist for those type of autoantibodies. An agonist is going to be an autoantibody that binds to a receptor and mimics natural ligand. And so it go ahead and we can do what we're supposed to do because the receptor got the signal. So it's like turning it on, it's amplifying it. This would be like Graves' disease, and that's why you see a hyperthyroidism issue. Hypoglycemia, another one. the insulin receptors constantly being turned on, saying, oh, there's glucose, there's glucose, there's glucose. We got to make more insulin, make more insulin, make more insulin. And then you end up with a super low blood sugar. Where an antagonist is going to block the receptor and not allow the actual ligand to bind. And so it doesn't activate the signaling, but rather shuts it down. And so we call, and so this would be like type 2, but diabetes. It's going to bind that insulin receptor, but it's not going to allow glucose to come in. It's not going to allow insulin to be made. And so you end up with, well, it's not going to allow insulin to be made, so then the insulin can't bring the glucose in. And so you end up with a hyperglycemia situation. So antagonists, agonists, depending on what type of receptor they're binding. A few other terms I want to throw out there are, when we talk about autoimmune diseases, we can use organ or tissue specific autoimmune diseases, where they're going to recognize one specific little part of the body, one specific receptor, one specific protein, and that protein is only found on one certain organ or tissue. A great example of this would be like Graves' disease again, where we have the thyroid epithelium is what is being attacked because that's where the protein is. Now we can have the alternate where an autoimmune disease can be widespread, systemic, all over the place. Type 2 diabetes, insulin receptors are going to be found on all the different nucleated cells. I think so, most of them. Maybe not all, but a lot, maybe all. I'm not even sure. Anyway, pretty much all. And all these cells need glucose to do cellular respiration. And if an autoantibody is going to bind to that, then it can bind to a cell pretty much on any tissue. And so it's systemic. There's also a handful of ways to treat autoimmune diseases, although they can't be cured. They can definitely be kept at bay, at least tried to for some of them. High doses of IVIG have been used with differing results. And an IgG that is given would bind to the IgG receptors or the FC gamma receptors on cells and prevent the actual, if an antibody is bound to a cell, it would prevent that, or a piece or a protein would prevent that from being taken. Anti-inflammatory response, which typically with a just putting in a whole bunch of non-specific antibodies can actually neutralize patients' own autoantibodies. It's kind of the theory behind that. It can downregulate antigen presentation. It can help with slowing down complement activation. It's a variety of different things. There's also the use of monoclonal antibodies again. So like TNF alpha. And so putting in a monoclonal antibody against something like TNF alpha can decrease the inflammation. And we see this helpful in a lot of inflammatory type of autoimmune diseases like arthritis, Crohn's, those kinds of things. Yeah. So what it really comes down to is that autoimmune diseases are difficult to deal with. They're complicated. They, can come up out of nowhere. Sometimes there is a familial association. Sometimes there isn't. Sometimes somebody can have the genetic predisposition for an autoimmune disease and never get one. So it's really, kind of an interplay between genetics and environment and fate And you just never know if somebody can end up developing one or not. And again, a major issue that we have with trying to figure out what drives an autoimmune disease, or at least what initiates an immune disease, is the fact that we don't know when it happens. When somebody actually starts having symptoms, the disease could have been progressing for a year, 2 years, and it's not like we can trace it back to one certain infection or one certain event in life. So it's very difficult to figure out where autoimmune diseases originate. All right, I want to tie together now these last 3 chapters that we talked about. This chapter and the previous 2 covered situations in which the immune system functions the way the immune system is supposed to function, but is doing it against antigens that we don't necessarily want an immune response against. And so it comes about by aiming cells and molecules against things that really pose no threat to human health. As a reminder, chapter 14 looked at allergies and how the immune response is inappropriate because it is mounting against things that don't matter. They're not a threat. They're just there, like things in our environment. In chapter 15, yes, we're recognizing non-self if we're looking at a transplant, but as humans, we put those non-self antigens into the body and we don't want an immune response to happen. And so these are given as a therapy to individuals who the benefit outweighs the risk of not having the antigen in there. And then in this chapter, we looked at self-attack. So although these are very different when we look at these diseases, allergies or transplant rejection or autoimmunity, they all still follow the same theme as a hypersensitivity, especially if we're looking at type 2, 3, and 4, where it can be antibody driven, it can be immune complexes destroying tissue, or it can be just cell destruction. But these are all different in their scope, but yet they all still have the same underlying kind of theme. So just want to tie that all together for you in this last slide. So hope you enjoy, and we'll see you next time.